গুড মর্নিং রে, একটু সবাইকে বল তাড়াতাড়ি জয়েন করতে।
Good morning, sir. Yes, sir.
Good morning, sir.
গুড মর্নিং বাবা, গুড মর্নিং। So, very good morning, children. We will start with the bacterial photosynthesis, as you all know. Now, we talked about photosynthesis in the last class. Now, what happens? We know that in primitive, in primitive bacteria, what happens? There are special type of small vesicles that are present, and these small vesicles are known as the chromatophores. Now, these chromatophores are roughly spherical in shape and has a diameter of 300 angstrom. So it possesses a diameter of 300 angstrom. Now, they contains they contains they contain what? They contain proteins and enzymes. Proteins and enzymes that are required for the process of photosynthesis. So it is possessing the enzymes and proteins that are required for the process of photosynthesis. Now, what happens? We know that in some bacteria there is presence of a pigment known as bacteriochlorophyll, and this bacteriochlorophyll is being essential. It is said to be as an accessory pigment, and there is an accessory pigment which is known as the carotenoid. So bacteriochlorophyll is the primary pigment or the essential pigment, whereas the accessory pigment is the carotenoid. Just write it down, Baba.
স্যার ব্যাকটেরিয়া ক্লোরোফিলটা কি বললেন?
Is the essential pigment.
আর অ্যাক্সেসরিটা ক্যারোটিনয়েড।
Carotenoid.
থ্যাংক ইউ স্যার।
Children, done. Children, finished. Should I rub it?
Yes, sir, finished.
तो बोल भी तो क्यों? Now, what happens when we talk about the photosynthetic pigments? Now, the photosynthetic bacteria, as I said, the photosynthetic bacteria can be of two types. One is the green bacteria, which is example is chlorobium, which contains chlorophyll C and has an absorption spectra at 650 nanometer, and also has chlorophyll D, which has its absorption spectra as 660 nanometer. Now, there is another type of bacteria being present, which is said to be as the purple bacteria. And this purple bacteria contains bacteriochlorophyll, bacteriochlorophyll, and it possesses an absorption spectra near about 800 nanometer. Please write it down.
স্যার,
হ্যাঁ বাবা,
ক্লোরোফিল সি এর পাশে ওটা নাম্বারটা কি লিখেছেন?
650 nanometer.
650, আর ক্লোরোফিল ডি 660।
Done, children?
Yes, sir.
Okay.
হ্যাঁ স্যার।
Okay. Now, what happens? The thylakoid, which are present within the cyanobacteria, thylakoid being present within the cyanobacteria is a bilayered structure, bilayered structure which contains galactoside, bilayered structure, and chemically it contains both galactoside diglyceride, right? It contains galactosyl diglyceride. Along with there can be along with one or more galactose molecule in the presence of in the place of the phosphate molecule of the phospholipid. So it containing what? In the place of this galactose molecule, it is containing, sorry, galactose molecule is being present in the place of phosphate molecule within the phospholipid. Is it clear, Baba?
As
Now, this thylakoid is responsible for what? For the conversion of light energy to biological energy into the cytoplasm. So it is responsible for conversion of light energy to biochemical energy. And where does it take place? It occurs within the cytoplasm. Please write it down. Done, children?
Yes, sir.
Now I will draw the diagram. Now what? How does it is looking like? Let's suppose this be the cytoplasmic membrane. What happens? This is the cytoplasmic membrane. This is the thylakoid. Now within the thylakoid, these are the reaction center. This is known as phycobilisome. Phycobilisome. And what happens? These phycobilisms are present facing cytoplasm. Now, what the importance of phycobilism? The phycobilism are considered to be the antenna molecule. Antenna molecule, which is responsible for the development of reaction center. Development of reaction center. So it is responsible for the development of the reaction center. Now, what happens? This phycobilism's contents phycobiliprotein, which acts as the motive force. Motive force. Write down.
স্যার,
হ্যাঁ বলো বাবা।
ওটা রিয়াকশন সেন্টার্স লিখেছেন?
হ্যাঁ বাবা, রিয়াকশন সেন্টার। ফিনিশ বাবা?
হ্যাঁ স্যার।
সবার হয়ে গেছে? ওকে। Now, what happens? We will talk about the green bacteria now. Now, the green bacteria, now what happens? The green bacteria, the chlorosome on the cytoplasmic membrane, there is presence of a structure known as chlorosome, which is associated, which is associated with the cytoplasmic membrane, right? Now there is what? There are presence of antenna molecules. So there are presence of antenna molecule. This antenna molecules acts as the base of the reaction. They acts as the base of the reaction. What I said, the antenna molecule acts as the base of the reaction. Now this chlorosome, chlorosome chemically comprised of chemically comprised of antenna molecules or chemically comprised of antenna molecules along with carotenoids. And what are they used for? They are used for the transfer of protons. Transfer of protons to reaction center. Just write it down over. ও যতক্ষণ না পাঠাবে ততক্ষণ তো স্যালারি অ্যাপ্রুভ হবে না না? Finish, children?
Yes, sir.
Okay. Now we will talk about another type of bacteria known as the purple bacteria. Now always remember this purple bacteria possess possess less developed photosynthetic structures. So they possess less developed photosynthetic structures than that of than cyanobacteria. Now, what happens over here? The intracellular membrane structure. Membrane structure contain the molecule and the reaction centers and the reaction centers. And this is a what? This is a This is a phospholipid bilayer structure. Write it down. Done, children?
Yes, sir.
Okay. Now we will talk about the just a minute. Now we will talk about the light reaction of the photosynthesis. Now what happens? The light reaction occurs number one by absorption of light by the chlorophyll pigment. Next, transfer of light from accessory pigment to chlorophyll A. And next is what? Transfer of transfer of energy by resonance and chlorophyll A to photosystem. So these are the systematic ways by which this transfer occurs. Next point is what? Excitation of chlorophyll. Next is what? Photolysis of water. Next is what? Electron transport. Please write it down.
স্যার, ওই অ্যাক্সেসরি পিগমেন্ট অফ ক্লোরোফিল এর অ্যারো দিয়ে ওটা কি লিখেছেন? ফটোসিনথেসিস অফ
Photolysis of
কি
water.
বলছে রাজশ্রী?
স্যার ওই অ্যাক্সেসরি পিগমেন্ট অফ ক্লোরোফিল ই এর পর যেটা আপনি লিখেছেন, ফটোসিনথেসিস অফ নাকি
Axillary
ফটোলাইসিস?
pigment of chlorophyll A, transfer of energy by resonance and to chlorophyll A. Next is what?
ওই
Photolysis. এটা হচ্ছে
অ্যারোড পর স্যার।
photolysis of water.
সরি।
Then হচ্ছে electron transport.
স্যার ফোর্থ পয়েন্টে কি লিখেছেন?
Excitation of chlorophyll. Next, photolysis of water. Electron transport.
থ্যাংক ইউ স্যার।
Now, can I rub it?
স্যার এক মিনিট
আচ্ছা।
লাস্ট পয়েন্টে লিখছি। স্যার হয়ে গেছে।
তন্ময় একটা পেপার রিভিউয়ের জন্য পাঠাবে, ঠিক আছে? ওটা তুমি একসাথে তো ওই আর একটা গ্রুপের যে কাজে তন্ময় স্পেশালি সেটা পাঠিয়েছে, তোমার কাছে রিভিউ করতে পাঠাবে। হয়ে গেছে বাবা? Okay. Now we will talk about non-cyclic photophosphorylation. And as over here, this will be for bacteria. You all have studied in plants mainly. Now, this we will talk of the bacterial cells. Now, what is the mechanism? That is P680. On excitation, goes to pheophytin. And these are what? Series of electron acceptors. Cytochrome B2F, and lastly it enters to plastocyanid. From plastocyanid it enters to what? That is P700, photosystem 700. Then what happens? That gets excited, from where it enters into again series of electron acceptors and results in the formation of NADPH2. H2. Huh? So these are the series of electron acceptors which are taking place from one electron acceptor to another electron acceptor. And always remember, over here the type of reaction which take place is oxido reduction reaction. So the type of reaction taking place is known as the oxido reduction reaction. Just write down. Finished, children?
Yes, sir.
All of you done? Rajashri হয়ে গেছে?
হ্যাঁ স্যার হয়ে গেছে।
Now, we will talk about another cycle known as the Kelvin cycle. Now, this is another very important cycle of photosynthesis. And in Kelvin cycle, what happens? We know the first electron acceptor is RUBP, that is ribulose 1,5 bisphosphate. And this ribulose 1,5 bisphosphate takes up water, carbon dioxide, and results in the formation of two molecules of PGA. Because we know ribulose 1,5 bisphosphate is a 5 carbon compound. When it accepts the carbon dioxide, it forms 6 carbon compound, and from where it breaks down to form 2 phosphoglyceric acid. Now this phosphoglyceric acid in the presence of ATP and NADPH2 gets reduced to form 2 glyceraldehyde 3-phosphate. This 2-glyceraldehyde 3-phosphate enters to changes to hexose phosphate, from where it goes to forms pentose phosphate, and this pentose phosphate is being formed by giving out of carbon dioxide, and this pentose phosphate is further recycled to form the carbon RUBP. Is it clear, children?
স্যার লাস্টে ডিসাইকেল,
হ্যাঁ বাবা,
স্যার
বুঝলাম না।
মানে প্রসেসটা কি বললেন, পেন্টোজ ফসফেট থেকে যখন আরইউপি চলে যাচ্ছে?
রিসাইক্লিং হচ্ছে তো
রিসাইকেল।
আবার। হ্যাঁ,
থ্যাংক ইউ স্যার।
ওয়েল।
So from hexose phosphate it becomes pentose phosphate in presence of CO2.
হেক্সোস ফসফেটটা পেন্টোস ফসফেটে কনভার্ট হচ্ছে বাই গিভিং আউট অফ CO2। কি, যদি CO2 থাকে তাহলে তো ওটা আরো বেড়ে যাবে কার্বন
Okay.
কন্টেন্ট, না? Done, children?
Yes,
Now,
sir.
okay. Now we will talk about another very important process. I won't be able to finish that, but I want to start with. That is nitrogen fixation. Now, what do we mean by nitrogen fixation? Anyone?
স্যার
This
প্লান্ট
nitrogen
ইউজেস,
fixation,
স্যার
just
নাইট্রোজেন,
hold on. The nitrogen fixation is what? It is the process of converting the atmospheric nitrogen to useful nitrogenous compounds. That is said to be as nitrogen fixation. Now, this nitrogen fixation comprises of number one, nitrogen cycle, nitrogen cycle, abiotic nitrogen fixation, abiotic nitrogen fixation, and biological nitrogen fixation. Now, this biological nitrogen fixation can be of two types. What is that? This biological, that is the biochemistry, or comprises of what? It comprises of the biochemistry of nitrogen fixation and genetics of nitrogen fixation. Just write it down over. Done, children?
Yes, sir.
Okay, so let me give the attendance first. Aditi, Aditi,
Yes, sir.
Ognivesh Sen,